Beyond kleptoplasty, the genetic integration of stolen chloroplasts in Elysia chlorotica. Imagine an animal that can eat a plant, and then become that plant, capable of soaking up the sun for energy. This isn't science fiction, it's the reality for Elysia chlorotica, the solar-powered sea slug. Bright emerald green, flat, leaf-like with lateral flaps, parapodia, that open wide like solar panels when basking. Adults range 3 to 6 centimeters long. Instead of one gut, it has an extensive network of branching digestive tubules that spread throughout the body. These tubules house the stolen chloroplasts, distributing them like veins in a leaf. Simplified compared to many mollusks, but perfectly suited for a sedentary, photosynthetic lifestyle. Typically around 9 to 12 months, often aligned with a single seasonal cycle of algae blooms. When E. chlorotica eats the filamentous alga Vorturia latoria, it pierces algal cells with its thular, a tongue-like rasping organ. It doesn't digest the chloroplasts. Instead, the chloroplasts are engulfed by specialized digestive cells. These cells protect the chloroplasts from degradation. The chloroplasts remain functional for up to 9 months, far longer than in the algae itself. This is puzzling because chloroplasts usually require nucleogenes from the algae to survive. Without those, they should break down in days. Studies, 2008 to 2010, suggested that E. chlorotica had stolen algal nucleogenes and integrated them into its genome, allowing it to repair and maintain the chloroplasts. Some thought to be related to photosystem repair, chlorophyll synthesis, and plastid protein transport. High-precision genome sequencing, 2014 to 2020, found no strong evidence of permanent algal DNA in the slug genome. Instead, the chloroplasts may be a preloaded E with enough backup proteins to last months. Or the slug might use epigenetic tricks or mRNA capture instead of full DNA transfer. Slugs behave almost like small green plants, spread out in sunlit shallow waters. Parapodia open equals maximum light capture. Parapodia closed equals resting, reducing desiccation or stress. Photosynthesis produces sugars and energy, sustaining the slug when algae is scarce. Can go nearly 10 months without food if sunlight is available. Still retains animal metabolism, so it uses both autotrophy, like plants, and heterotrophy, like animals. This makes E. chlorotica a mixotroph, both plant and animal at once. Challenges the line between kingdoms of life. Suggests early evolutionary pathways where multicellular animals might have once experimented with plant-like strategies. Provides a real-world example of symbiogenesis, two organisms merging functions, similar to how mitochondria and chloroplasts themselves originated billions of years ago. Some scientists consider E. chlorotica a modern echo of ancient endosymbiosis. Each individual has male and female reproductive organs. Deposited in ribbons, often coiled around algae. Hatch without chloroplasts, appearing grey-brown. First feeding on Vorturia latoria ingestion of chloroplasts, body gradually turns green. Adults may stop feeding altogether, surviving purely on sunlight. This transition makes its development look like an animal turning into a plant. Found in salt marshes, shallow lagoons, tidal pools, 0 to 0.5 m depth. Camouflage, looks like blades of seaweed or algae, protection against predators. Predators, few documented, possibly due to toxic or unpalatable compounds absorbed from algae. Seasonal life, populations rise and fall with algae blooms. Understanding how foreign organelles survive in animal cells may advance gene therapy and organ transplantation. Studying this symbiosis could inspire artificial photosynthetic systems. Offers insight into how major leaps like endosymbiosis could occur. From its vibrant color to its stolen genes, Elysia chlorotica. remains one of the ocean's most captivating mysteries, reminding us that nature still holds endless secrets, waiting in the shallow waters for those curious enough to look.